So you grab a diet soda or maybe one of those sugar-free energy bars, right? And you've got to wonder, what exactly is making this taste so sweet without bringing the calories along for the ride? Well, do you know exactly how much artificial sweetener is actually hiding in your zero-calorie snacks? Today, we are going to find out by literally putting those exact treats under the microscope.
Welcome to today's Explainer. I am absolutely thrilled to guide you through a comprehensive virtual lab masterclass, Detection of Artificial Sweeteners. We're going step by step, from a raw food sample straight through to final, undeniable data.
I'm your host, Dr. Kim, lead scientist, and honestly, my absolute passion is breaking down complex analytical chemistry. I want us to understand exactly what's in our food, and more importantly, how we can scientifically prove it's there. So let's get our lab coats on. Okay, here's our syllabus for today's masterclass. 1. The science of sweetness. 2. Essential lab materials. 3. Prepping our samples. 4. Running the detection procedure. 5. Crunching the numbers. And finally, 6. Real-world applications.
Let's kick things off with Section 1, the science of sweetness.
Before we even try to isolate these molecules in the lab, we've got to ask, how do these chemicals trick our biology? Well, artificial sweeteners actually interact with a super-specific sensor right on your tongue. It's a G-protein-coupled receptor known as T1R2/T1R3. Think of it like a biological lock, and these sweeteners, they're the master keys. They trigger a signaling cascade that tells your brain, hey, this is sweet, even though they deliver absolutely 0 calories. They are, quite literally, chemical illusionists.
And because of that biological trickery, we see some wild structural diversity among these compounds. You've got everything from asulfamate K and saccharin to sucralose and aspartame. I mean, they have completely different molecular shapes, yet they all somehow manage to unlock that exact same sweet taste receptor.
Now, because their structures are so incredibly diverse, there's no single magic bullet for detecting all of them in a food safety lab. Take aspartame and saccharin. They have these things called chromophores, which are parts of the molecule that absorb UV light. So boom, easy to detect via standard UV spectroscopy. But then you have sucralose and cyclamate. They completely lack chromophores. They're practically invisible to UV light. To catch those, we have to step up our game and use advanced tools like mass spectrometry or evaporative light scattering detection.
All right, moving to Section 2, essential lab materials.
Let's gather the tools of the trade. To pull these sweeteners out of crazy complex food matrices, like, say, an energy bar packed with proteins and fats, we need some specific reagents. We're talking methanol, acetic acid, and ethanol. Crucially, we need Carez solutions number 1 and 2 to force interfering proteins to precipitate out. Add to that our 0.2 micron PVDF filters, solid phase extraction cartridges for a really deep cleanup, and aluminum-backed HBTLC silica gel plates for the visual chromatography.
Which brings us to Section 3, prepping our samples.
Whether it's a diet soda, a thick dessert, or even human plasma, we have to clear out the biological noise. Here's the workflow. Step 1, dissolve and sonicate, using sound waves to break down aggregates and degas the liquid. Step 2, Carez clarification. We drop in those Carez solutions to crash the proteins right out of the matrix. Step 3, centrifuge and push the liquid through our PVDF filter. And finally, Step 4, the solid phase extraction cleanup. This brilliant little SPE cartridge acts like a trap. It catches our target sweeteners while we wash away all the remaining junk.
Down to the fun part. Section 4, running the detection procedure.
Our samples are pristine, so we load them into the LC-MS/MS. That's liquid chromatography with tandem mass spectrometry. This setup is unbelievable for detecting multiple sweeteners all at once. We start by spiking the sample with internal standards. Then, chromatographic separation spreads the molecules out over time. As they exit, electrospray ionization charges them up, followed immediately by MRM detection, which just zeroes right in on the exact mass-to-charge ratio of our target sweeteners.
And when we use HPTLC, we get these really fascinating charred spots on the silica plates. These spots are actually sucralose that we isolated from a traditional Indian dairy dessert called basundi. Now, what's mind-blowing here is that basundi is processed at intensely high temperatures. We're talking up to 115 degrees Celsius. These dark, distinct spots are visual proof that the sucralose doesn't degrade. It remains perfectly stable and intact, even after enduring all that heat.
Let's look at the math. Section 5, crunching the numbers.
Because, visual proof is great, but a true scientist always backs it up with hard data.
For standard soft drinks using UV spectroscopy, we rely on good old Beer-Lambert's law. Big A equals little a times B times C. Put simply, by measuring exactly how much UV light our sample absorbs, we mathematically calculate the exact concentration of the sweetener.
But when we're dealing with those complex extractions through SPE and mass spec, we have to use this master equation. The concentration in micrograms per gram equals Y times V times D times W, all divided by M times Z. It looks like a lot, right? Let's break it down.
Y is just the concentration the machine actually reads. V is our extractant volume. D is the dilution factor. W is the final volume. Then we divide all of that by M, the initial weight of the food sample, multiplied by Z, the volume we push through our SPE cleanup. The big takeaway here? By relentlessly tracking every single drop and gram throughout the lab process, we translate raw machine outputs into incredibly precise dietary exposure levels.
Which brings us home. Section 6, Real-World Applications.
Let's step out of the lab for a second. Why does all this dense chemistry actually matter? Well, look at the matrices we tested. For low-calorie basundi, our HPLC methods proved the sweetener's heat stability, which tells manufacturers it's safe to use for extending shelf life. For human plasma and urine, LC-MS gives us the extreme sensitivity required for actual clinical and toxicological studies. And for your everyday soft drinks and candies, UV spectroscopy helps regulatory bodies verify that the labels are telling the truth. Ultimately, this exact science helps resolve the ongoing debates about the health impacts of these additives.
Which leaves us with one pretty massive question. As food manufacturers keep inventing entirely new, complex, zero-calorie substitutes, what undetected compounds might be hiding in our future diets? Well, to figure that out, we're just going to have to head back into the lab. Thank you so much for joining this Masterclass Explainer. Keep questioning and keep analyzing.